Today, we are going to tell you about one example of modeling work going on at the Institute. There are 4 of us presenting this talk, but this is actually a much bigger team effort, so many more people have contributed. And all 4 of us are from the Modeling, Analysis and Theory department at the Institute, but we work very closely, as you will see, with the experimentalists here. And also I have to say that we will have time to show only a few pieces of the data and of the analysis and the simulations that we have done. There is much more to it that we don't have time to show you, but please come ask us questions if you are interested. So why are we talking about the visual physiology of the cortex in silico here? So, well, in silico is simple. That's because we are doing simulations in silicon on a computer. But regarding the visual physiology, we are talking about it here because that's what's being done experimentally. A majority of our in vivo experiments are visual physiology. And the main purpose of most of the modeling projects here at the Institute is to be very closely intertwined with the experimental interrogation and to try to establish this virtuous cycle of experiments and modeling. And what do I mean by that? Well, what we are trying to do in many of our modeling approaches is to integrate as much as possible of the experimental information that's being collected. So we try to take the information about characteristics of individual cells and cell types and about their connectivity, as well as results of in vivo recordings, such as those that you heard about from the previous team talk and those that you will hear more about in the last team talk of today. And so we try to combine all that information in comprehensive, specific models. So we don't want to build abstract models of just some kind of networks. We want to build models that are very specific for this part of the brain that we are studying. And so in that way, we use this information to drive this modeling effort at several levels of resolution. And ultimately, what we want to do is to be able to compare our modeling simulation results as directly and closely as possible with the experiment. So that, as you will hear in this talk, involves in particular trying to use the conditions that are as close as possible to the experimental reality, and in particular the visual stimuli. So because a lot of what we do experimentally is, as I said, visual physiology, and as you heard in the previous team talk, basically that involves showing various types of movies to the cells, to the mice, and then analyzing responses of cells to them. We need to do the same in the simulation, and that way we'll be able to compare directly our results to the experimental results, and ultimately we hope that this will lead us to predictive models such that we'll be able to run our simulations, find interesting mechanisms that underlie the activity that we observe both in the experiment and in simulation, and being able to direct future experiments to make discoveries. And so in that way, we'll be able to close this loop, the virtuous cycle of experiments and modeling. We are not quite there yet. This is a long process, and we are doing our best. And today we'll tell you about one example of how this is being done, focusing on one particular approach, the biophysically detailed network modeling. And here, our model that we have built over the last few years has been the model of the input layer of the mouse primary visual cortex, the layer 4. So this is the part of the primary visual cortex that receives direct inputs from the primary optical pathway. And so to build this model, we took advantage of our Allen cell types database, which involves characterization of individual cells in slice. So we use those models of those individual cells that have been developed as building blocks. When we did this several years ago, we didn't have too many cell models to work with, so we chose 5 that were the best out of what was available back then. And so as I show here, they represent a few of the putative cell types, excitatory cell types in orange, magenta, and red, and 2 parvalbumin fast spiking interneuron types. So we take the cells and we replicate them many times to create this model that's shown here, that contains 10,000 biophysically detailed cells. So now these are cells that have very sophisticated description of the biophysical mechanisms of their ion conductances, and they use multiple compartments to represent the morphology of cells. So these are very detailed, computationally expensive models. So we have 10,000 such biophysically detailed cells. They are surrounded by another 35,000 of much simpler leak, integrate, and fire point neuron models that provide boundary conditions. And this whole model topographically represents the middle portion of V1 and corresponds to a little bit more than half of the layer 4 in V1. And then on top of that, we add recurrent connections, which is, of course, an important feature of the cortex. And for that, we use some of the preliminary information from our ongoing in-house experiments, but also relied very heavily on the literature. And finally, we distilled all that information into the connectivity rules. So basically, the probability of connections between different cell types depending on the distance between their cell bodies. So those rules were applied. And so now we have cells, we have connections between them. And what is left is to provide inputs. And the primary input into the layer 4 is from the primary optical pathway. And those inputs are necessary to connect this model to the visual stimuli. And so now you will hear from Ram how this has been established so that we could drive our models with the visual stimuli and what happens once we establish that and we're able to compare to the experiments.
Thank you, Anton. So as Anton mentioned, we need to develop a model for inputs to this layer 4 model that we are developing. And we know that visual information does not reach layer 4 directly in the form of a movie. Rather, information gets processed in the retina and the LGN, and then this information is passed on to layer 4 in the form of input spikes from the LGN. So in order to enable this transformation from visual stimuli to spikes, we developed a simple filter-based model of thalamic inputs. Typical spatial and temporal filters that were used in our thalamic models are shown here. As a first approximation, each LGN unit was modeled as a spatio-temporally separable filter, and the spatial and temporal kernels used are shown here. As a first approximation, we used 9,000 such LGN units consisting of 3 functional cell classes. These are the simple cell-like on and off subunits and the more complex cell-like on-off subunits. These units were connected to layer 4 using simple retinotopy-based connectivity rules. Once we had this model, we could then use it to transform any arbitrary visual stimulus to spikes, which can then be used as inputs to our detailed biophysical models. So we used this thalamic model in order to be able to generate responses of layer 4 neurons to a wide variety of visual stimuli and compare model responses with experimental data. So shown here are spike rasters for responses of all 10,000 biophysically detailed neurons in response to one particular drifting grating in our stimulus set. The plot in the middle shows example tuning curves for one representative excitatory SCNN1A neuron and one example inhibitory interneuron, which is a PV1 interneuron. In agreement with experimental data, the excitatory neurons in our model show sharp orientation tuning, and in comparison, again in agreement with experiments, the inhibitory neurons show relatively flat orientation tuning curves compared to the excitatory neurons. The panels on the right show comparison between model cell classes and experimental data for 3 different metrics that can be obtained from neural responses. The top panel shows the spontaneous or background firing rates of all neurons in the model. The middle panel shows the maximum response in response to drifting gratings, and the bottom panel shows the orientation selectivity index, which acts as a measure of sharpness of such orientation tuning curves. In all these cases, the colored bars represent model cell classes, whereas the gray bars shown here correspond to experimental data. As can be seen from these plots, the model shows reasonably good agreement with experimental data in almost all cases. In the case of the interneurons, the orientation selectivity for the interneurons, the model does not show as good an agreement, and we are currently working on refining both our thalamic input model as well as the layer 4 model in order to obtain better improvement in this case. In addition to drifting gratings, we also show you here responses of model layer 4 neurons in response to a natural movie stimulus. So shown here on the left is a 3-second clip from the movie Touch of Evil, which is the same movie as was used in the Allen Brain Observatory. Shown below are spiking rasters of the biophysically detailed neurons in response to this 3-second movie clip. As you can see from the raster plot, the responses to natural movie, in agreement with experiments, the model shows temporally precise responses. In addition, the variability of responses for the different trials also goes down in response to natural movies in agreement with experimental data. On the right is a 3-second clip of the same movie. However, in this case, the different frames of the movie have been scrambled randomly. And in this case, in contrast, when you look at the spike rasters, you see that the responses are not as temporally precise. And in fact, the responses in this case resemble more closely spontaneous activity as opposed to the more detailed structure that you see in the plot on the left. Finally, also in agreement with experiments, responses to natural movies also evoke sparser responses, as shown in this plot. This plot shows you the fraction of cells having a particular lifetime sparsity, which is defined according to the equation on the right. The different terms in this equation have been explained here. However, intuitively, one can think of this metric as representing the following cases. In the event that this number is high, this implies that the cells are way more responsive and way more selective in responding to only particular frames of the natural movie. However, when this number is a little lower, it means that the cells are not as selective in their responses in response to the different frames of a movie. So this plot shows, in agreement with experimental observations, that natural movies produce more sparser responses as compared to drifting gratings. So with that, I'll hand it over to Yazan, who will tell us a little more about some mechanisms underlying model responses and some predictions from our simulations.
Thank you, Ram. So one of the things that we wanted to study is how the different mechanisms of, and in particular, the underlying architecture of the recurrent connectivity affects the overall dynamics of our network. And one of the reasons we wanted to study this is because of different types of functional connectivity that have been measured in the literature. And by functional connectivity in this talk, what I'm referring to is not what's commonly considered where the correlation of responses between neurons is defined as functional connectivity, but how the architecture relates to the functional properties of the neurons. And so one example of this, and this is all data that has been measured in the literature, and this is all for layer 2-3, is that the architecture is designed in such a way such that A, neurons with similar orientation tuning are more likely to be connected to each other, and B, more strongly connected to each other. So given these two kind of constraints, we wanted to include that into our network architecture and see how that will affect the overlying activity. And these are some simulations to show. So actually the red curve is when both conditions, higher chance of connectivity and stronger connectivity of similar orientation tuning cells is imposed, while the black curve is completely random, and that constraint is not imposed. And the green and the orange are kind of either or, using only one of the features. And we can see that to get orientation selectivity indices that are comparable to the literature, which is what the red curve is, and the values are close to what the literature reports of 0.5 to 0.6, we need to impose this functional connectivity on the network. And this is interesting because, one, it tells us a mechanism for the network of how we can get strong orientation selectivity, and we need to impose the recurrent connectivity rules, and it's not just going to happen by the feed-forward connectivity. And two, this is a model for layer 4, and so it kind of makes a prediction that we would expect similar architectures in layer 4, because as of right now it's only been seen in layer 2, 3 of the mouse, and we're kind of excited that maybe experiments in the future will test this, and we'll see what the experiments will say. And using the model, we can then also try and understand other mechanisms from other experiments that have been done. And so this is actually data that I'm going to be showing that's recorded by our very own Sean Olsen. And the experiment is such that Archer was expressed into SCNN1A neurons, which is one of the cell types of the 5 types that were modeled in the network. And by shining a light, those cells can be inhibited. And at the same time, there's full recordings happening along the V1 column. And what's shown on the far side is the responses for one cell. So every row is a different trial of a stimulus. And in this example, the stimulus was a grating, which was applied when this black bar is on. And the red trials, kind of the color coding in red, is for the trials where the light was being shined, and hence the cells were being silenced, or the SCNN1A cells were being silenced, and this particular cell as well was also being silenced. And so we want to quantify this for every cell and kind of look at the overall population of cells, and this was done with an optogenetic modulation index. And the general idea is that if it's negative, that means that cell's activity decreases because of the silencing of the SCNN1A. And if it's positive, that means it's an increase in activity because of the experimental perturbation. And so what we wanted to see is compare the simulation with the experiments. And so the simulation is shown here in the black curve, and the experiment is in the gray curve. And what we can see is the simulation qualitatively captures some of the features, or the features of the experiment. And so we see that it very nicely captures that there's a fraction of cells that are actually silenced completely. At the same time, there's another kind of bimodal distribution, and there's a population of cells that seem to be right around 0 plus or minus a little bit, but kind of not really affected very strongly by the experimental perturbation. But one of the cool things about having a model is that we can go and interrogate this a little bit further, and we can look at the 5 different cell types individually and see how they're all affected. And so this is kind of what's being shown here, a plot for all the 5 different cell types in the model. And we can see that actually the cells that are being completely silenced are the SCNN1A neurons. And they're the ones that we are silencing, that have been silencing experimentally. But at the same time, we see that the other populations, they're the ones who are the major fraction that's not really affected. There's a slight increase in the other 2 excitatory cells, and perhaps a slight decrease in the inhibitory cells because they are receiving less excitation from the SCNN1A neurons. But we can start to look at the mechanisms of how the perturbation experimentally and then replicate it in the model is affecting the different kind of features of the model. And another kind of great feature of the model is that we can then go and silence different populations individually and see how that affects the overall network dynamics. In the interest of time, I won't be able to show those results because right now we've kind of been showing how we provide an input to the model and we see the output, and we now want to go in the reverse direction. Given the output of a model, what does that tell us about the input of the model, and comparing that with experimental results? And for that, Michael will introduce that topic.
Thanks, Suzanne. Okay, so this is a model of a sensory system. And of course, in order for a sensory system to be useful to an organism, it has to transmit information about the external world. So one way you can assess whether or not that's happening is by doing a decoding experiment. So what do I mean by this? I mean, what we want to do is construct some function which tells us about how the stimulus choice is manifested in the cellular response or the spiking response of the particular model. And then you want to use that to see if you can take a given response and reproduce the actual stimulus. Now, given that we're talking about, in sort of, for example, one case, one of our simple stimuli is drifting gratings, you might think this is very easy if you've opened your textbook and you see tuning curves, because tuning curves are often drawn like these nice, clear things, and you might think, oh, I only need a couple of cells which have tuning preferences that are slightly offset, and then that would be very easy. But of course, this is not quite so easy, because if there's neural variability, neurons do not respond the same way every single time. You wind up with something like this. And so you have to somehow pull information out of a set of responses like this. And so the question becomes, how many cells do you need, and can you do this? Now, what I'm going to do is show you examples of doing this in a couple of different ways, one of which uses a decoder that just uses the tuning curve. So it averages this response over trials and treats all the neurons as independent units. It doesn't actually correlate or use the information in the correlated activity. The other kind of decoder actually uses the fact that maybe the green cell will fire above its tuning curve sometime, at the same time as the blue cell fires below its tuning curve. Those are called noise correlations, and they can possibly also carry information. So, Yazan just described to you several different instantiations of this model which have different kinds of connectivity, depending on whether the connectivity is like-to-like or random, or depending on whether the synapses are like-to-like or random. So I've grouped these different, the decoding results for these two different decoders. The solid bars are the independent decoder, and the hashed bars here are the correlation-based decoder for the different types of architecture. Now the interesting thing right here is that for the drifting gratings, which are on the left here, the sort of realistic architecture, if you will, produces a lot more information or transmits a lot more information, whereas in the other cases they don't relative to that. And this doesn't seem to matter for the natural scenes. Now I set up these decoding experiments in the same way as our brain observatory to sample the same kind of section, so a 400 micron view in some layer, so that I could in fact compare this to doing the same decoding results or do the same decoding experiment as the brain observatory. The thing that jumps out at you, or one thing that might jump out at you, is that you get maybe similar results for the rate model decoders, but you do not get similar results for the correlation-based decoders. This suggests that the tuning curve or the rate properties we're actually capturing probably reasonably, but we're not capturing the correlated activity. And with that, I will return you to Anton.
Thanks, Michael. All right, so as you've seen already with this relatively simple model of just the layer 4, we are able to capture a number of experimental observations, and we have many more that we didn't have the time to show you. There are also things that we don't capture so well, and these are important. This is the learning experience which will help us think and improve our model in the future, which is something that we are doing. And then another thing that we are doing is we're actually extending this model to describe a bigger and more complex portion of the brain in this model. Namely, we are now building a model that would cover almost the whole cortical depth. Instead of just the layer 4, we have the layer 2, 3, layer 4, layer 5, and layer 6 in this model. And so we are also working with a much more advanced version of the Allen cell types database now, which contains many more models of individual cells. And using that, we are able now to distinguish multiple cell types per layer. And for each cell type, we actually using also multiple models of individual cells. So that in the end, now we have built a preliminary version of this model of the almost full V1 in the mouse. And it contains more than 50,000 biophysically detailed cells, about 250,000 cells altogether, because it again has this sea of point neurons for boundary conditions. And it includes about 20 different cell types and 112 individual cell models. And so we introduced connectivity to the system, we introduced external inputs from the LGN, we were able to run the simulations of this model, and it runs pretty well. This is a very computationally expensive model, but we have shown that we can run these simulations. Now it needs to be optimized to make sense. And then once that is done, we hope to perform a number of virtual experiments like those that we just showed to you for the layer 4 and compare to the experiment. And eventually we hope that models like this will help us to make predictions that could be tested experimentally and we hope would lead to discoveries. And with that, we'll finish and thank all our colleagues for their multiple contributions and our founders for their help and support. And thank you for your attention.
Okay, let's take a few questions. I'd like to warm up with one for Yazan. So you hinted at different roles for the different inhibitory cell types in your model, and I wonder if you could expand a little bit in the context of the inactivation or other experiments about what you think they might be doing, again, either for the intrinsic dynamics of the network or for possibly for a stimulus representation. Why do we need so many?
Yeah, that's...
that should take the full 5 minutes.
There's a lot of speculation that can be done there. So in this particular model, there was only the parvalbumin cells modeled, and even though there were 2 subpopulations. And we know that the diversity of the inhibitory neurons is quite vast. And the main big 3 that people like to think about is probably the PV, the SST, and the VIP or HTR. And there's been some disinhibition kind of effects being shown. So an inhibitory neuron will inhibit another one, which is effectively exciting the 1-2 synapses down. Overall, what's the magic number to kind of give each one a specific role? It's a difficult, it's already a question that would be nice to get an answer to and would require a lot of experiments. I would say the inhibitory neurons have a bunch of roles, such as one, there's been like some results showing that they're important for contrast invariance. So if you change the contrast of a grating, you still want the excitatory cells to vary, and a part of that is brought in by the inhibitory neurons who will kind of modulate things such that they can still be as responsive and also gain modulation. So one, and a little bit on the speculative side, but one kind of role for the inhibitory neurons would be to, one idea would be that they are there to kind of allow the brain or the visual cortex in this particular area to respond to a wider dynamic range by kind of allowing, so not just adaptation on a single cell level, but adaptation on kind of like the whole network level. But that is definitely like a longer conversation with a chat.
Very good, very good. And we have another question in the front here.
Yes, I wanted to discuss this result. So you showed this lifetime sparsity with respect to movie frames, which was puzzlingly high. So my question here is that if we think about what we know about V1, which has orientation tuning and retinotopy and so on, why would a V1 cell have specific tuning to a given movie frame? If we look at the primate brain, we clearly see such cells, but not, they would probably be found in IT. So that's a really interesting cell.
Well, I think that in this particular case, our movies are very short, those that we sampled, and there is only a few of them. And so I don't think it's so much, this high sparsity is not so much the result of this particular cell being selective to a very particular image, like, a face or something, but more that in the limited number of samples that we show that only one of them had the combination of edges that was, exciting this particular cell. So I think, I wouldn't read too much into the exact number of the sparsity values. The more important result for us is that we clearly see the difference between the natural movies and the gratings or other types of stimuli like that.
I have one question to the comparison between the models and the data. You compare this on a spiking level, no?
Yep.
Do you plan also to do that on a sub-threshold level? Because I can well imagine that there are many models that give you the average firing rate, right? But that look completely weird on the sub-threshold level. Do you plan to do that in the future, or do you already do that?
Well, ultimately, we would like to. Here at the Institute, we don't have such a big effort on measuring membrane voltage activity in vivo. We have a little bit, but not a huge pipeline. And so we haven't looked into it very extensively yet. But yes, this is certainly a very important thing for us, especially because these are biophysically detailed models, and they are supposed to give us exactly that level of resolution.
Very good. Let's thank this team one more time.